Today we're going to be doing chapter 15, which is regulatory mechanisms in animals from Heinemann 1. And these are the questions that you need to look at doing. There's only 9 of them, and they shouldn't take you too long to get them done either. We're going to start off by looking at homeostasis. And homeostasis is the ability to maintain a stable internal environment despite changes in the external environment, so changing the conditions which that organism is in. So that organism wants to keep everything inside of itself running exactly the same regardless of what environment it is in. And it uses hormones and it uses nerves to be able to maintain those things. And here we're relying on negative feedback systems. So negative feedback systems are where the response that is occurring because of the stimulus reduces the effect of that stimulus. So because this stimulus, this sign, this signal causes a change within the animal, and the animal then responds to that change, and the response to that is going to reduce the effect of that stimulus, that sign, that signal. And in other words, so the fancier words is the response provides feedback that has a negative effect on the stimulus. So the stimulus that we're talking about here, this is the thing that signals the change. So it's a change in something. So the stimulus could be heat, touch, smell, sight, any one of those kinds of things that how we detect changes. And we use receptors, and these receptors are what actually are able to detect those changes. So these are able to detect the stimulus. And when it detects this change, it then sends a message to the control center. And the control center that we're really going to be talking about here is the brain. These control centers decide what type of response we need, how strong that response should be, and if we even need to respond to it at all. So if we touch something and it's painful, this is a quicker response, but generally we then decide that we don't like this pain, so we need to remove ourselves from this painful situation. So once the control center has made up its mind on what it's going to do, we then go to the effector, and this is the thing that makes the change within us. So the stimulus says this is what's going on, our receptors detect what's going on, our brain says, all right, this is what's happening, so we're going to make these changes, and the effector are the changes that are responding to that stimulus. So the effector are the thing that changes the stimulus. So if we're cold, for example, the stimulus would be a cold environment. The receptors would be our skin receptors. We're going to be detecting that it's cold. Our control centers are going to say, okay, we're getting really cold here. It's too cold for us. Let's try and use some of our mechanisms that we have to maintain internal temperature. And then the effectors are going to be doing that. So some of those might be for shivering, for example. So the effector there is going to be our muscles, and our muscles shivering to generate heat. And then that response restores the original state that the receptors then pick up again, send the message to the control center saying, yeah, that shivering's worked, our body temperature is now rising, so then the effector is able to turn off. So it's able to stop you from shivering. So they work around in a circle motion continually, and then these stimuli come in from time to time to change that receptor response. So if we're looking at temperature, we're looking at either being hot or being cold. We're talking about vasodilation if we're hot, sweating, pilorelaxation, and stretching out. So if you are hot, you're going to dilate or make bigger the capillaries that are closest to the surface of your skin. So if this is the surface of my skin and I have a capillary, I'm going to make that capillary bigger because then it's able to hold more blood, and then from that I'm able to release the heat into the environment because it's closer to the surface of my skin. For sweating, that is where you're then going to have this water molecule with salts and other ions on the surface of your skin. And then when that evaporates, that's going to have a cooling effect. Pilorelaxation is where the hairs on your body are going to lay very flat, and you've got stretching out. So laying down, like stretching your arms away from your body, separating your legs. So really laying in a starfish kind of shape so that you're able to create a really large surface area for this heat to leave you. If you're cold, you're basically going to do the exact opposite to all of those things. So you're going to be doing vasoconstriction, shivering, piloerection, and curling up. So instead of those arteries getting bigger, we're going to constrict them. We're going to make them smaller. So we're going to take the blood from the surface of your skin and move it to your core to keep your organs warm. Shivering, this is going to generate heat. So it's a very rapid contraction and relaxation of your muscles to generate heat. You've got piloerection, so the hairs on your body are going to stand up. This traps air around the hair that is then able to absorb the heat from your blood vessels that are close to the surface, and it creates a warm layer of air close to your body. And then you've got curling up, so making yourself as small as possible so that you reduce the surface area that is in contact with your environment, and therefore you should lose less heat. So we're going to have a look at the hormonal systems that we have now. This is one of the ways that we're able to maintain our environment. So these hormones are found in all animals. The hormones are a diverse range of compounds, and they act as intercellular, so between cells, intercellular messengers. So they send a message from one cell to a different cell, and they usually have a slower response when compared to the nervous system, and they target particular cells. So they're intercellular, they're messengers between cells, and they have a slower response than our nervous system. So we've got different types of hormones, and we're going to start off with the steroid hormone, which is also known as the fatty acid hormone, and it is lipid soluble. Remember, lipid meaning fat, and soluble means it's able to dissolve. So steroid hormones are able to dissolve into fats, which means that they're able to enter the cells. Remember that phospholipid bilayer. So they're able to dissolve into the fatty membrane layer around the cells to then enter the cell. They bind specifically to receptor molecules within that cell, and here we're looking at things like testosterone and estrogen. So these are two hormones that we have for the male and female reproductive system. So you can see with this picture on the right, here we've got our hormones, our steroid hormones. They're released from the capillaries, and then they're going to go through the membrane layer because they are lipid soluble, and they're able to go into and attach to these hormone receptor complexes, which can then signal something else happening within the cells. So for example, estrogen is going to help with our production of eggs inside of females, and testosterone for the production of sperm. Another one that we have is non-steroid hormones, which are also known as amino acid hormones. These ones are water soluble, so they're able to dissolve into water but not into fat. And these require receptors outside of the cell because they're not able to go through that cell membrane. Remember, it's a fatty layer and they're not able to pass through it. And here we're looking at adrenaline and your human growth hormone, or HGH. So this picture here shows the hormone as the blue polygon that we've got here. And it's going to attach to the receptor molecule, which is this one here. So when it attaches, it activates something inside of the cell. In this case, it's going to activate that G protein. So once that hormone attaches, it's going to activate that G protein and cause that change within that particular cell. We've got some glands that secrete most of our hormones, and here these are our endocrine glands, and they're a cluster of cells that excrete hormones. So they produce the hormone and then release it, and where they release it to is the circulatory system, because that's the quickest method for these hormones to transport around the body. So if we're going to look at the pituitary gland, this plays a central role in regulating the endocrine system. It is a hormone that regulates other glands, so other endocrine glands. So once the pituitary gland is triggered, it's going to release a hormone, and this hormone is going to trigger a different endocrine gland to release a different hormone. And it works with the aid of the hypothalamus. And you can see by this picture here that it's up inside of the brain. So what we're going to be doing in class is we're going to be looking at creating a life-size model on some paper. Okay, now let's have a look at the nervous system. So the nervous system is a lot more quick in terms of its response when we compare it to the hormonal system. It is highly specific and it does require energy for this to happen. So each nerve cell is made up of a neuron, N-E-U-R-O-N, and these sensors, there are different sensors on cells that pick up on these messages, and these neurons must transmit their message to an effector cell. Remember, we talked about those cells being the ones that have that response to the stimulus. So you detect something and then you can have a nervous system response to that stimulus. So here you can see the body of the nerve. So here you've got your body of the nerve cell, here you've got your axon, and you have your dendrites as well. So inside of the nerve, we've got our axons, which are the longest, the long tube that we were talking about previously. And inside each nerve are many nerve cells. Okay? And you've got veins and you've got arteries supplying the blood with the oxygen and everything else that we need. Because remember, nerves do require energy, so it means we do need that oxygen. Connective tissue, here we're looking at muscles around the nerves to protect them. So while we're looking at one nerve, it's made up of heaps of nerve cells. So previously I said that the nerve cell was made up of the cell body, and then you've got the axon part to it as well. So each of those axon long strips of the cell have one of these axon bodies. So each and every single one of these little circles that you can see, the little orange circles, that's a separate nerve cell within a nerve bundle. So here you can see the cell body that I was talking about before. These dendrites are able to connect to different things, so say muscles or anything like that to detect it. The nucleus is in the center of the cell body. You've got the axon, which is the length of that cell that the message is going to travel around. It's covered in a myelin sheath, so each of these little oval type of circles is a myelin sheath, and between them is a node of Ranvier. And then you've got your axon terminal bundles at the end, and then between this nerve cell and the next nerve cell is a synapsis. It's a gap between them. So what happens is the message needs to move along this nerve cell. So the axon will run through the middle of the myelin sheath, and you've got those nodes of Ranvier between each of those myelin sheath. The nerve impulse doesn't travel through the axon, it actually jumps over the top of those myelin sheath and makes contact with the axon. And when this happens, potassium ions leave the axon and sodium ions enter. So K plus leave, Na plus enter. And then this nerve cell won't be able to pass another message again until the potassium ions are back inside the axon and the sodium ions are outside of the axon. So it's able to monitor and control these messages using sodium and potassium. So another word that we have here, we've got our stimulus, we've got our nerve impulse, which we can also call action potential. You'll hear it referred to as that a lot. So the stimulus is detected by the dendrites. The cell body then pushes this nerve impulse, that electrical impulse, or that action potential through the axon to the axon terminal bundle. Here we've got chemical transmission. So each of these little nodules is then going to release a chemical across the synapses to trigger either the next nerve cell or whichever cell it's attached to. So reflex responses, these are rapid and unconscious responses, but they do still rely on nerve cells. So things like pain, so excessive stimulation to a sensory receptor, and these use fast pathways. So here the brain isn't necessarily involved in making these decisions, it's more the spinal cord that decides what happens. So if you prick yourself with a pin, which is what's happening at the bottom here, you've got your receptor, your pain receptor, and the message goes down the nerve cells to the spinal cord. So you've got your receptor, the sensory neuron that's going to pass the message, the integration center inside the spinal cord, and then that's going to decide what happens. Then this message is then going to go to the motor neuron, because we don't want to be touching something that's painful. So we send it through a motor neuron, which then has the effector in muscle cells to then remove yourself from the pain. So sometimes we don't need the message to go all the way to the brain, just to the spinal cord. And this is why the spinal cord's really, really, really important in helping us being able to move and make decisions and those kinds of things. So we've got our central nervous system. There are different areas to it and they have different functions, and the brain coordinates all of the responses from every other area, and it basically is the storage point of all of this information. So we're going to be looking at the brain stem, your spinal cord. You've got intercostal nerves, which are ones that run along your rib cage. Femoral nerves are ones that run down towards the inside of your leg. You've got your sciatic nerve, and you've got tibial nerves, which will run down towards your toes. So we've got two different types of systems within the central nervous system. We've got the somatic and the autonomic, and then within the autonomic we've got the sympathetic and the parasympathetic as well. So if we're looking at the peripheral nervous system, this is what moves us towards the central nervous system. So the peripheral includes your sensory nerves and motor nerves that are located external to, say, your brain and those other nerves that I was discussing before. So this isn't a part of your brain, this just gets messages to your brain or to your spinal cord or to your femoral nerve. And it's divided into the somatic and the autonomic. So the somatic transmits messages from the central nervous system to your skeletal muscles. So from your spinal cord to your muscles, and this is things that are going to help you to move. So skeletal muscles are the muscles that we use to be able to move. The autonomic nervous system transmits messages from the central nervous system to smooth muscle, heart muscle, and glands. So these are things that we don't have control over. So like things like our heart, we don't have control over when our heart is beating. So I can't just think, oh, I need to remember to beat my heart. My heart is going to keep doing it without me needing to think about it. It does it when I'm asleep, does its job 24 hours a day, and it responds to things such as exercise. So the autonomic nervous system does things without us controlling it. So your smooth muscle, so for things like breathing, your heart muscle, and your glands as well. So here we've got the sympathetic and the parasympathetic divisions of that autonomic nervous system. So the parasympathetic, we're looking for things like sight, stimulation of saliva, a slow heartbeat, the constriction of bronchi, stimulating peristalsis inside of your esophagus and then your stomach and those kinds of things, releasing of bile, and the contraction of your bladder. So those are all parasympathetic. And then you've got your sympathetic, which is your pupils dilating or becoming bigger. It inhibits or stops the flow of saliva. It accelerates your heartbeat, so it makes your heart beat faster. It makes your bronchi dilate and become bigger. It stops peristalsis. It converts glycogen to glucose, has a secretion of adrenaline and noradrenaline, and inhibits bladder constriction. So we've got some major sense organs, and these systems really all rely on our sense organs detecting changes in our environment. So we've got our eyes, ears, nose, skin, and tongue. We've got photoreceptors, chemoreceptors, and mechanoreceptors. So photo being light, chemo being chemical, and mechano being movement receptors. We've talked about the action potentials before when we looked at the structure of the nerve. So the action potential is that electrical impulse that travels along the length of the axon. So what happens is it's responding to a stimulus, and what it does is it depolarizes. So it goes from being negatively charged to positively charged. And if that depolarization, if that positive charging is big enough, that action potential is then generated. So it starts off with a negative charge, it starts to get positive. As that positive charge builds, once it hits that threshold, that particular point, that action potential generates and that message has to be sent. If it's below, you're not going to get an action potential. It's either, yep, we're going, let's get this message out there, or nothing's going to happen at all. So it's similar to do with once you've decided to do something, it has to happen. So if you've decided to try and catch a ball, then you're going to go and try and catch that ball. There is no changing your mind once you've made that decision. That action potential message has already been sent to your muscles, for example. So what happens is the channels open and sodium floods into the axon. At a certain point, those sodium channels are then going to close and the potassium channels inside the axon are going to open and the potassium is going to leave. Those potassium channels are then going to close, and then this sodium-potassium pump is going to restore that balance between potassium and sodium back to its original level so that it is again negatively charged and able to then complete several more action potentials. So this particular nerve cannot be used again until it is restored back to its negative charge and the sodium and potassium levels are restored to their levels, because that is what it's relying on for this action potential message to be able to be passed down the axon. So here what you can see is that we start off with the negative 70 charge, and then here we're starting to get our time charge on the left side here. And as we go across here in more time, you can see here that this initial signal here wasn't big enough to necessarily stimulate the exact response. As soon as it hits this dotted line here, that's the threshold for action potential. So as soon as it hits there, then the sodium is going to start entering the cell. The potassium then leaves the cell to balance it out again. So we start to get slightly more positive. The sodium then floods in once it hits a certain point, and then the sodium leaves that cell, so making it more negative. And then this is where that sodium and potassium pump really comes into play, because it's going to take it back to this resting potential that we had before. So if we're looking at an action potential, this is inside of the cell here. You can see the graph up the top is exactly the same as the one we had on the other side. And down the bottom here, it's describing what happens. So 1, at negative 40, the sodium channels open and the sodium channels flood inside. So these sodiums are going to go inside. At plus 50, these channels then close and the potassium channels open so that the potassium can then leave the cell. So the voltage, because of these potassium ions leaving, then drops to negative 90, and those potassium channels then close. And then this pump is going to restore the sodiums and the potassiums to wherever they were before. So we generate a nerve impulse by our sensory neurons. So it's the conduction of an impulse along those axons is how nerve cells communicate with each other and with other cells. It's the chemical transmission of a signal to another cell across a synapses. Remember that synapses is the gap between those cells. And you can see it here in this final picture. So we've got our action potential traveling along the axon. Axon terminal means the end of the axon. So we're at the end of the axon. We've got our action potential going down here, arrive at the axon terminal. Number 2, here coming into it, we've got calcium. So the positive calcium channels open, calcium enters the cell, and it's going to signal these synaptic vesicles. And these synaptic vesicles contain the chemical message. So that it signals the vesicles. These vesicles then move to the membrane. So that's step number 5. They then dock to the actual cell membrane, and then via exocytosis, those chemicals are then released into the synapses. So you've got your synaptic cleft. So they're released into the synapses between the two cells, and then on the new cell is the receptor, and those chemical neurotransmitters attach to the new cell. So we've got our action potential calcium comes in, those synaptic vesicles attach, releasing the chemical neurotransmitter across the synapses, which then attach to the receptor to continue the message or to finalize their response or to send their message further.